morning, students. Welcome to BSc 2 Zoology class. So today I am going to discuss on human development up to implantation. This we already discussed. We have been discussing. So today we shall see it in detail. So development of human embryo. See, after fertilization, now look here, from ovary ovum is released. At the time of ovulation, the released ovum is in secondary oocyte stage. It is in secondary oocyte stage, means it has not yet completed its second meiotic division. In the second meiotic division, at metaphase 2, its growth is stopped. At that time only, the ovum is released from the ovary. That release of ovum from the ovary is called ovulation. Okay? So ovulation occurs very well in middle of the menstrual cycle, the ovum is released. If the menstrual cycle is of 28 days, on 14th day, more or less, on 14th day, the ovum is released from the ovary. That is called ovulation. After ovulation, the released ovum is captured by fibria of one of the fallopian tubes. Later on, it enters into the fallopian tube. Fallopian tube is lined by ciliated epithelium. Ciliated epithelium is there. So these cilia beat. They beat to push the ovum towards the uterus. Okay? This is ampullary region, isthmic region. Between ampullary isthmic region, the ovum is moving. That region is called as fertilization site. At that time, the sperms are entering into the uterus and fallopian tube after the intercourse or coitus. After copulation, the sperms are released into the vagina. From the vagina, they enter into cervix. From the cervix, they enter into uterine cavity. From uterus, they enter into fallopian tube. Very few thousand sperms may enter the fallopian tube. At this region, fertilization takes place. Fusion of male and female gametes. Details of fertilization we already discussed. So after fertilization, soon after fertilization, what happens now? Soon after the entry of sperm into the ovum, second meiotic division is completed. As a result, first polar body is released. Second polar body is released. So at the end of first meiotic division, first polar body is released. At the end of second meiotic division, second polar body is released. Okay? Now, the male pronucleus and female pronuclea both are fused. This is called as the karyogamy or amphimixis. Soon after that, zygote is formed. So zygote has two sets of chromosomes. It is diploid. Whereas this one is second oocyte is haploid. It is haploid, this one is diploid. This diploid nature is due to the fusion of male and female gametes. Okay? Male gamete sperm brings 23 chromosomes. Female gamete ovum brings 23 chromosomes. Totally it would be 46 chromosomes, right? So diploid number of chromosomes of human beings is 46. Now the zygote undergoes cleavage, division. So first cleavage occurs around 30 hours after fertilization. The first cleavage takes place along the central axis from animal pole and vegetal pole axis. Okay? So first cleavage is in human beings holoblastic and slightly unequal. Slightly unequal. One blastomere is smaller, another is larger. Okay? So at the end of first cleavage, two cells are formed, two blastomeres are formed. Right? Next, second cleavage occurs at right angles to the first one. As a result, four cells are formed. Four cells are formed. Okay? Later on, second cleavage onwards, the planes of cleavage are irregular and they occur at different planes. They occur at different planes and several cells are formed. Now, morula is formed. Morula stage. Morula may contain around 100 cells when it reaches uterus, uterine cavity. It is still externally covered by zona pellucida. Can you see this? Zona pellucida is there around the morula stage. So, fertilization, cleavage, morula stage. Morula is multicellular embryo. It doesn't possess cavity at the center. Okay, it is a hard multicellular ball-like structure. Right? Later on, these cells start to secrete secretions at the center of the embryo. As a result, what happens? Cavity is formed at the center due to the release of secretions from the peripheral cells. See here in the uterus, by that time, the uterine wall secretes secretions called uterine milk. That uterine milk is rich in nutrients. This milk is sucked by the peripheral cells. These peripheral cells of the morula. As a result, they are larger and they are bigger in size and they are getting flattened. They are flat. So these cells are known as trophoblast cells. And at the center, cavity is formed due to the accumulation of fluid secreted by surrounding trophoblast cells. Okay? So here what happens is, cavity is there and this cavity is covered by flattened cells. This layer is called as trophoblast layer. At one region, small mass of cells is formed and this is attached to trophoblast. This mass of cells, inner mass of cells, is going to be developed into embryo, actual embryo. The surrounding trophoblast is not taking part in formation of embryonic body, but it helps in formation of extra embryonic membranes like chorion, amnion. Okay? So this part is called as inner mass of cells or embryonal norm. Okay, so this embryonal norm is attached to it is in contact with the peripheral trophoblast cells, and these trophoblast cells are called cells of Gruber. Cells of Gruber. Okay. So now what happens? Due to the absorption of uterine milk, the size of the embryo is increasing. Clear? As a result, what happens? The pressure is being created on the wall of zona pellucida. Now zona pellucida ruptures. Right? As long as zona pellucida is present around the blastocyst, this stage is called as blastocyst. Okay? Blastocyst. So this cavity is called blastocyst. Clear? This blastocyst doesn't attach to the wall of the endometrium, wall of the uterus, as long as it is covered by zona pellucida. When zona pellucida is ruptured, the blastocyst is released into the cavity. Now this is able to adhere to the wall of the uterus. Now it adheres here. it attaches to the wall of the uterus. So wall of the uterus has been divided into three parts, three layers. Outermost layer is called as perimetrium, very thin layer. This is perimetrium, and inner layer is middle layer is called myometrium, and innermost layer is called endometrium. Endometrium. So this endometrium is very important as far as attachment of blastocyst is concerned. Now blastocyst attaches to the wall of the uterus, that is endometrium. So this attachment of the blastocyst to the wall of the uterus is known as implantation. Implantation. Okay, so implantation is a process wherein blastocyst attaches to the wall of the uterus. Right? Now, see, this attachment of the embryo starts about 7 days after fertilization, and it takes another 3 days time to complete the process of implantation. Complete the process of implantation. Okay? Now, this is attaching to the wall of the uterus. Where exactly, where the inner mass of cell is there, that part is adhering to the endometrium. Now at this region, the trophoblast cells start to secrete enzymes. These enzymes rupture, erode the lining of the endometrium. As a result, what happens? Finger-like projections are sent into the tissues of endometrium. These are called chorionic villi. What are these? Chorionic villi. So these finger-like projections enter into the endometrial tissues to suck the nutrients and other requirements from the maternal circulation. So later on, it gets embedded into the endometrium. So endometrium encircles the blastocyst. Okay, so around the blastocyst, the chorionic villi develop to get the food from the maternal blood circulation. Chorionic villi, and the uterine tissues are interdistated. Right? See, after implantation of blastocyst, the endometrium. So endometrium becomes decidua. Decidua is shedding part of the uterus during implantation, parturition. So decidua is nothing but endometrium. Okay. So now the decidua has been differentiated into three types, such as decidua basalis. Decidua basalis is the part of the endometrium where the embryo is attached. This part, decidua basalis. Then the decidua or endometrium, which is present around the embryo, is known as decidua capsularis. Decidua capsularis. Second one. And decidua paritalis is this one. This is not connected to the embryo directly. Okay? In between decidua capsularis and decidua paritalis, there is a cavity. This is uterine cavity. What is this? Uterine cavity. Decidua paritalis. So thus, there are three regions of decidua. Decidua basalis, number 1. Number 2, decidua capsularis. Number 3, decidua paritalis. So this is cervix, vagina. So the embryo is completely implanted, embedded into the endometrium. So for the complete process of implantation, it takes around 3 days time. 3 days time in human embryonic development. Okay. So this is about the implantation of the human embryo. So with this we have completed completed the developmental biology part that is given in your syllabus. So in the next class onwards, we shall see the physiology part. Thank you.